Okay, now presenting, we have Jeffrey Meckler with Indaptus Therapeutics.
Thank you, and thank you everybody here and also listening on the webcast. At Indaptus Therapeutics, our mission is pretty simple. It's how we can use and engage the immune system to cure disease. I will be making forward-looking statements, so I do advise everyone to see our latest filing. So in a nutshell, we are a company that has a unique platform, and we are a clinical company. We entered the clinic this year with our first dosing of our first therapeutic. And I'll go through what our technology, but just as a little overview, what we are is a company that has a platform that really develops therapeutics. These therapeutics come as a package of immune agonists. And in simple form, really what that means is we engage the immune system and we use that as a way to go after different diseases. Our first foray into that will be oncology. We're really excited in the fact that we initiated our first trial this year, and in addition, last week we were at AACR, which is one of the premier cancer research conferences, presenting some new data, and I'll actually have a couple slides from that data we presented. So what is this technology? So essentially, what we do is we're based on this concept, and historically, people have seen like cancer and other diseases get cured in the presence of an infection. This led our founding scientists to come up with the concept of, well, what if we could engage the immune system in a way that would kind of replicate this but do it safely? So he came up with the technology, and it's a bacteria-based platform. And what we do with that is create a therapy that kind of emulates that. We call it Decoy. It fakes the body out in the thinking there may be something that the immune system needs to turn on for. But we do it in a very safe way. And the reason is we're able to just kind of pulse this multi-targeted agonist approach. What we do that's unique is we have shown single-agent tumor eradication as well as synergy with a bunch of different, multiple types of current therapies. We've seen both the induction of innate and adaptive immune systems, and we've shown immunological memory. So let me kind of explain this in a way. What current therapies have done, and I'll use the pointer here if I can. You don't really see it. What current therapies have done is target a single part of the immune system. The concept has been, oh, this one switch, that will cure cancer, or that will make all the changes we need. And so what you've seen develop over the course of the last few years has been, whether it's checkpoint inhibitors, CAR T, targeted antibodies, they're all focused on one approach. Well, what we're doing is we're actually looking at engaging all of these components in a way that can naturally use the immune system to fight disease. And the reason this is very important is because, as it turns out, single approaches have not been as effective as people thought. And what we're seeing people experiment, when I say people, other investigators and companies come up with therapies where they're starting to combine it. We're going back to how Mother Nature, how the immune system was designed, and engaging it. So what we've developed is really this multi-packaged pulse-primed approach. And it really starts going back over 100 years to a doctor in New York, William Coley, who came up with an idea. And it was really by observation of how these therapies existed. And what he did was he saw people with bacteria infections, and they would be cured of their solid tumor cancer. So he came up with a killed cancer kind of therapy. And it was very toxic, and there were a lot of issues in trying to figure it out. Now, what happens is you fast forward in the last 20 years, we've learned a lot about the science and the technology of why this would work. And what we've done is engineer a platform around that. Now, one of the questions I often get is, well, I don't hear about this happening today. Well, the reason is we invented antibiotics. So now when you have an infection, you immediately are getting treated with antibiotics. Your immune system is not fully engaging as it would years ago. That's why you don't see as many spontaneous regressions as you did, say, 100 years ago. So kind of the nuts and bolts of this is we start with this engineered bacteria. And what we now know is lipopolysaccharide, LPS, is the most toxic component in gram-negative bacteria. So what we do is we remove about 90% of it. By removing it, we're now making this safer. So now we have this package. One of the posters that we had at AACR last week, we actually showed for the first time the characterization of our first product. And we show we have all these different toll-like receptors. We have NOD, we have STING. Essentially, we have all the different switches to turn on different parts of the immune system. So what is this? This is a single strain, non-pathogenic gram-negative bacteria. We reduce lipopolysaccharide. We process this in a way it's given as an infusion, as an IV therapy. What's really interesting is this is passively targeted to the liver and spleen, and it has a short duration in our animal studies, and as we go into humans, we'll validate that, but short duration within the system. So we're excited, and this year we became a clinical company. We dosed our first patient. We're in the process of really understanding the safety profile of these new therapeutics. That's really the main component of this year. As we get in the latter part of this year and in the next year, we'll then start in our expansion phase of our trial and then go on to our combination study. So what you're going to see as an investor over the course of this year and next year is more data on the patients, the safety profile, the types of immune profiling we're able to do in humans, all the human data to really justify how this therapy will work. We'll then move into the expansion, and when we get in the expansion component of the trial, that's where we'll start giving it multiple doses and replicating what we see in some of our animal studies, which I'll show you shortly. So we dosed this in the first quarter of this year. We'll have a lot of safety data coming out in the second half of this year, and then we'll start our multi-dosing here, and we'll have the multi-dosing safety data next year. So I'm going to, for this presentation, since this is really more a generalist, not a scientific, I'll go through some of these slides, but I'm going to keep it kind of at a high level and not go through too much. This slide here basically compares the toxicity level, this is the endotoxin activity, between a non-treated the bacteria and our therapeutic. And it shows basically we've reduced much of the toxicity in these assays. This slide is interesting. It compares a couple therapeutics that we had developed from our technology, and it basically is on the far left. You see, by taking out the toxicity, the lipopolysaccharide, we actually did not reduce the activity in several different anti-tumor cytokine immune responses. So this is great. We reduced toxicity, we kept some activity. And on the right side here, what we do is we compare that to traditional single agent activity. So single toll-like receptor 7/8, you see activity, we see much more significant. So what we believe is that that will be indicative that you'll see greater anti-tumor activity in the immune system. This is something that was published last week at AACR. It basically is showing that we have these different toll-like receptors. So what we're giving is a package of immune activators as well as NOD and STING. Essentially, it's just confirming what we always believed. As we process around the periphery of the gram-negative bacteria membrane, we are capturing these immune agonists. So this is something you've probably seen a lot if you've looked at different companies that are in cancer. And basically, we take a mouse and we give it a metastatic pancreatic carcinoma and tumor. And then what you see is within about 30 days, that's that first kind of line, all the mice have essentially died. And we then go and give them some gemcitabine, which is a drug that's usually the backbone in pancreatic cancer therapy today. And it basically doubles the survival timeline. Now, this is an early study, so we gave some different dosing of a Decoy therapeutic. And what you see is we actually almost extend that out another 20-some days, almost 30 days. So what we're actually seeing here is we're seeing single agent activity. What's really interesting here is for the first time, and again, this is a new slide, new data from last week, is we're actually this is a breast cancer model. We actually show single agent activity, but 2 out of the 5 mice in this study actually had complete regression, and it was durable out to 90 days. What that means is the mice were dosed with our therapeutic, and not only did the tumors not grow as fast, actually in 40% of the mice, the tumors completely regressed and disappeared. So we are showing single agent activity and single agent tumor regression, which is something very rarely ever seen in these models. So here's another model. This is an hepatocellular carcinoma, essentially liver cancer. And again, just with adding an NSAID in this model, we would get 50% of the mice complete response. This, they don't like using the term cure in cancer, but this is complete regression. They have no more cancerous tumors. Now, this is a study that was done, and I like this slide a lot because it shows what the model, if you do no therapy, these tumors just grow. That's that blue line. The green line here, that's just showing you what if you give the state-of-the-art, best-selling cancer drug. It's an anti-PD-1. Decoy has very similar profile, but when you combine the two, 100% of the mice have complete response. 100% of the mice have their tumors totally regressed, and 5 out of the 6 mice at day 91, which is a very long time in mice studies, are still durable. So we essentially cured 80-some% of these mice of cancer. I'm just going to move through a couple of these things. One of the key attributes of our therapy, which is not seen very much, is what's called immunological memory. When you get a cold, a flu, a virus, you get sick, but then you have, the next time you're exposed to it, your body has a memory. It can actually attack that immediately. So what we did was we took mice from one of the earlier studies that had completely regressed, and we gave them another tumor. This was, I believe, hepatocellular. And we re-challenged them. So we gave them the same cancer tumor again. 100% of the mice, when re-challenged, completely regressed. And again, you can see it was durable out past 90 days. So we are showing that our therapy not only is synergistic, not only can we have cancer eradication, but we can also have immunological memory if that exposure reoccurs. Even in today's, the best-selling drugs with checkpoint therapies or CAR T, there is rebound, there is reoccurrence. We're showing this in our preclinical models that we can actually address that issue. What is also very unique with our therapy is we're showing this type of activity in multiple types of tumors. So this one is non-Hodgkin's lymphoma tumors. Again, you see immunological memory, you see the tumor regression. Now, one of the questions that we get from the real sciencey people sometimes is, wow, that's great. We've cured mice of cancer 100 times. What about humans? We have started our human trials, but this is a model where we're actually using human cells, not mice cells. So this is a human tumor xenograft model. Again, in the model, when you add Decoy to what is low-dose chemotherapy and Rituxan, again, you're seeing 100%. This is synergistic, and it makes sense because we reset the immune system. We really are teaching our immune system to get back to doing what it's designed to do. This is a slide that shows that we can induce immunological memory and the innate immune system. I won't go into too much detail because that gets a little scientific-y. One of the things, before I go for some questions, I want to really state that this is a platform. Oncology, cancer is our first venue, but we believe this will have application into any immune-mediated therapy. So here we have shown data in hepatitis B. Hepatitis B, there is no cure. Current standard of care, entecavir or tenofovir, people go on it, and when they're on the drug, the virus is undetectable. As soon as they come off the drug, the virus comes back. They get the scarring of the tissue. This leads to loss of liver function, liver cancer. What we think we can do is including this into a therapy where it'll be a combination. It's improving the immune response with other therapies. We've also actually shown that we have activity in HIV, and we're working in other models as well. We think once we get our safety data this year, there's an opportunity for us to look for other ways to utilize the platform. Lots of people like to talk about, well, how big of a market is this? How are you going to go approach it? So far, 6 of the world's 12 deadliest cancers, we show activity and this works. We believe that over time, we're going to be able to make several different therapeutics out of our platform that will address different things. Whether it's different tumor types or different viruses, it won't necessarily be the same set of immune agonists. So we will create different versions of our therapeutics to address different cancers, different viruses, whatever. And this is just showing you that in the market opportunity for us is huge because we could go after, these are cancers we've already demonstrated activity. We always get asked about our management team. This is a very lean virtual style company. We have 6 employees. The management team here, as you can see, our backgrounds. And this is a team that has worked in big pharma, small biotech, international, domestic. We have a lot of activity that we are able to kind of process because of the senior management. But even more impressively is our board of directors. We have a number of people who have led these types of, I'll call them almost revolutionary. This is a new modality. This is a different approach to how to go about fighting cancer and other diseases. Michael Newman, who is our founder, who invented this, he was actually in charge of cancer biology at Novartis. We have Brad Hayes, who is the CFO of LabCorp. We have our chairman, Dr. Pomerantz. Roger was the founding CEO of Ceres. He was in charge of licensing for Merck. And we have Mark Gilbert, who was the chief medical officer and brought one of the first CAR-Ts on the market at Juno. We just recently brought on Dr. Martel, again, at Tesario, bringing one of the early CAR-T therapies to market. So with that, I think I have a few minutes left. I can open the floor to questions or can finish the presentation. Yes. Sure. The question is, I mentioned CAR-T therapy. Is that what we're doing, and what is the difference? So what CAR-T therapy is basically taking, harvesting cells from your body, reprogramming them, and reintroducing. What we're doing is something very different. What we're essentially doing is tricking the body into having an immune response. And one of the things that happens when you have cancer is we now know, and this is research over the last couple decades, that the way the cancer evades the immune system is it actually systemically reprograms, it resets the immune system. By having an event that kind of turns these switches back on is what helps the immune system go after the cancer. So what we're essentially doing is trying to trick the body into thinking there's a reason to reset and turn back on. Yeah, well, the bacteria, so our therapy, we start with a bacteria and we kill it. We process it, but it's the cell membrane's intact. These are intact. Think of it as an intact cell therapy, but it's a bacteria. But it's gram negative. It's non-pathogenic. The bacteria that we use actually can't replicate in humans. It needs an enzyme that humans don't have. So on top of it, we have multiple safety components in there. So, but the body then sees this cell and interprets it as a bacteria and then activates. Did I answer the? Okay. Sure. Let me. I'm thinking if I can bring that up. Maybe, maybe not. I think it's right here. So that was our cash and cash equivalents at the end of last year, according to RK. We are now I advise investors don't look at the historical. We were not burning a lot, but now that we started the clinical trial, we'll kick up. We're probably burning a little under $1.5 million a month. So our cash runway takes us into Q2 of 2024. Does that answer your question? Yes, sir. So the question was, what is the scope of the first clinical trial and the challenges? So our first clinical trial is an all-comer solid tumor dose escalation trial. So what this means is we really are taking kind of the sickest of the sick to come into our trial. They've had to have failed prior therapy. They're coming into our trial as a somewhat last resort. And this is a pretty sick crowd. I very often have to advise investors that, the expectations are we're looking for safety. We think we really have to get into the multi-dosing and the combination to really see the power. And we think long-term, as you develop this is probably something that'll move earlier patients as a therapeutic. To get into clinical trials in this area, this is the path forward that the FDA has, is that, we can, try this. This is a new modality, and these patients are very sick. We actually bring them into a hospital setting for the first set of these patients. They spend 48 hours in observation. We actually observe them for 28 days post. So did I answer the question? Yeah, I would say that's a fair way. It's a conservative approach, yes. Well, if there aren't any other questions, I thank everybody and thanks for coming to the conference.